Hello, my name is Jaleigh Goben, and I'm a PhD candidate at Cornell University. I am a paleontologist by training, but I apply my expertise towards modern conservation issues, a growing field called conservation paleobiology. More specifically, I study the recent history of oyster reefs in the Guana River in northeast Florida, which is part of the Guana River Marsh Aquatic Preserve that was established in 1985. Recent research projects have also provided a better understanding of how current shellfish impact water quality within the Guana River Estuary. However, little is known about long-term impacts beyond the last 10 years. My research is working to extend that baseline further into the past by accessing the preserved record of accumulated dead oysters beneath living reefs. I've collected 5 to 10 foot long vibrocores of living reefs across Guana River and have been measuring the size of oyster shells and counting abundance throughout these cores to identify how the oysters have changed over time. Over the last 5 years working on this project, I've experienced firsthand how special the Guana River Estuary is. Not only has it become a meaningful place to me, but I care about its future for the plants, animals, and people that call it home. The Guana River Estuary has been labeled an outstanding Florida water body, meaning it is worthy of special protection because of its natural attributes, and is considered impaired due to excess nutrients. As a result, eutrophication and harmful algal blooms threaten the environment, species, human health, and local recreation within the estuary. Oysters offer a natural solution to mitigate excess nutrients through their water filtration capabilities. A single oyster can filter up to 50 gallons a day, which helps to remove sediment, algae, and other pollutants from the water. Local resource managers are currently considering whether adding oysters could help improve water quality in the Guana River and have been monitoring the oysters since 2014. The goal is to use oyster size and density from the past to quantify how much filtration Guana River oysters were capable of before 1985, when the aquatic preserve was established. I can then compare these past estimates to how much the current oyster population can filter to determine whether adding more oysters to Guana River is feasible based on the recent history of oyster size, density, and filtration. Generally, the deeper a shell is within a core, the older it is. To determine how old the shells are and how much time is represented in my cores, I've been using a method called radiocarbon dating, which measures the carbon content within an oyster shell to estimate the year it died. Preliminary radiocarbon dating indicates oysters from pre-1985 are accessible within the cores I collected. However, more radiocarbon dating is needed to accurately reconstruct the recent history of Guana River oysters. This can be costly at $165 per shell. Not only would this project inform management decisions, but it would also provide a pilot study for how to apply the historical oyster record to fill in data gaps in other areas where oyster reefs exist. Thank you.